Welcome to the second episode of Journal Walk. So today is a windy day. I may have to pause the video, stop and restart many times just to avoid the noise. But without further ado, let's get started. So today I'm going to talk about a paper from Chaudhuri et al. In Lafayette's lab. It's published in 2019, just last year, in Nature Neuroscience. You know, in system neuroscience research, especially those trying to understand the neural underpinning of behaviors, you know, we usually use like tasks. We design a behavior for the animal to perform, and with this designed behavior, we have many behavioral variables that are of interest, and then we find candidate brain regions and drop electrode into those brain regions, and to see what kind of activity we get, and if we can decode those behavioral variable we design or not. But you can see an obvious problem with these kind of approach, right? So the brain may be encoding a lot of other useful things, and those things also about the task and the performance, but the decoding is usually biased. You know, it's targeted to those variables, not taking into consideration of other potential information. But it's hard to do any better than those, right? Like back in the days, even still now, for a lot of studies and in a lot of labs, then we can only record a handful of neurons at the same time. Earlier it was like one neuron at a time. It didn't really have the statistical power to resolve more information in an unsupervised way. As a result, people might have just thrown the baby out with the bathwater. So, as Simo Benzer said, progress in science is usually a result of the interplay among new technologies and the discoveries and the new ideas, probably in that order. In the past 2 decades, especially in the past 10 years, we have seen a boom of new technologies. We have developed a new electrode that can record up to thousands of neurons at the same time to get a single unit activity. But with a bunch of data stored on a hard drive in the form of numbers, it's really hard to make much sense out of them, right? So in my opinion, we need to bring some new math, new in quote, into the neuroscience research. And when I say new math, I didn't really mean, although that includes that, but I didn't really mean like Isaac Newton invented calculus to address his physics problems. But I'm more talking about that the math that's already exists in mathematical research, and or even widely used in the other fields like math, physics, but it's still kind of unknown or new to neuroscience research, or at least is only narrowly adopted in a small part of the community. For example, topology is one that I think is super powerful on addressing the neural network related questions, and also to a lesser extent the analytical approaches about the dynamical systems. In Simo Benzer's quote, people sometimes didn't emphasize enough about the keywords interplay between technology discovery and the new ideas, rather than they kind of see it as a succession. I think the paper I will be talking about, it really links the rich data we get from the advancement of technology, leading that to potential new discoveries that we can make out of those data. Furthermore, I think it may even lead to a paradigm shift of how we analyze this kind of rich neural data. Then we kind of reverse the process. So it could be a design, or it could be a very natural behavior, and we record those activities, and then we try to identify the activity structures in an unsupervised way and independent from any artificial variables. And then we have certain structures, and we ask ourselves, like, what kind of behavioral variables and processes can account for those activity structures, and how and furthermore how those variables can be useful or informative for solving certain tasks and controlling the behavior. Specifically in this paper, the authors chose the head direction system. What is a head direction system? Like in the brain, there are some neurons that are sensitive to certain direction that the head is oriented to, and this sensitivity is not with respect to my body, where my body is facing, but it's with respect to the landmarks of my environment. So you can first imagine I have a ring of neurons surrounding me, and when I'm looking straight, the neuron in front of me is firing, and when I turn my head to the camera, then the neuron in the direction of the camera is now firing. And similar for other directions as I turning my head. Well, actually, in insects like flies, there is physically, literally a ring of neurons in their brain, and then representing different head directions. In mammalian brain, and here in this paper we're going to mainly deal with anterior dorsal nucleus of the thalamus. Those head direction cells are not organized in a ring, but their firing activity and can be seen as, at least from theoretical models, is situated in a ring of the old activity space, and this is called a low-dimensional manifold. So in addition to the manifold structure, the model theories have proposed that like this head direction circuit is a line integrator. So when we say integrator, that means the system has memory, right? The input can, so let's say we are at certain states in this space or at a certain point on the ring, and there are inputs telling the velocity of where the head is turning to. Well, not turning to, like the velocity of the head turning, and the velocity are transient. For example, I'm turning my head from here to here. That's a transient velocity input, and that transient velocity input can get into this head direction circuit, and as a result, the state will be shifted to a new state along this manifold, but it will stay there even the velocity input is gone. So it's memorize that, or in another word, it's integrating the input along this manifold. Okay, now we can jump into the actual result of this paper. But again, before we get into the main method, we need to do a little bit of topology 101 to understand the procedure. So Betti numbers are ways to measure the topological features of certain structures, of certain geometric structures. And the Betti 0, which is the first Betti number, describes how many isolated entities are there in the space. And the Betti number, the Betti 1, which is the second Betti number, describes how many one-dimensional holes are there in the space. For example, if the space has one ring, the ring has a Betti 1 of 1 because it has a one linear hole in the middle. And if you think about a torus has two one-dimensional holes in it, one along the torus and one in the middle of the torus, and it also has a three-dimensional hole or void, which is inside the torus, inside the ring of the torus. And if we're recording from 20 neurons or 50 neurons, then we are dealing with a 20 or 50 dimensional space. And at every moment in time, we have 20 or 50 firing rates, and then those firing rates give us 1 point in the space. And if we record for a prolonged period of time, then we have a lot of samples at a different time point, and then that gives us a cloud of points, or point cloud. So what it did to identify the main structures in those cloud points is by growing bubbles out from the individual points. Just to think about it, if you grow bubbles from two adjacent points, and when the radius of those bubbles become large enough, the bubbles will merge together and form a new identity, right? So, and that will cause the Betty zero to be changing, right? Because the number of isolated entities are changing. Or imagine in some circumstances, then we suddenly have a ring of bubbles merged together, and then all of a sudden that will give us a new feature that has a Betty number of 1. And here we see what's called what the authors called the Betty barcode. So here shows as the bubble grows and the radius increases, so we started to have many smaller rings, and then but we know like the point of clouds are organized in a ring, and therefore for a large range of radius, and we have a ring structure which is indicated in yellow. And so therefore, using this method, and the authors could identify something called a persistent homology, and it's a persistent topological features during this process. And that persistent feature reflects some true, or is likely to reflect some true structures in the activity distribution. So once we identified the dimension or the topology, which is a one-dimensional ring, and then we can the authors fit a spline to this, a spline with dimensionality of 1, and then the authors parameterize this spline with a variable here called alpha. However, for the observed data, and they are probably not exactly on this spline, but in order to assign the alpha values to the original firing rate or states, then the authors simply take the shortest distance between those points to the manifold, and then therefore to read out what is the corresponding alpha value it is. And here let's look at the actual data. So the data is recorded from mice, awake mice, just exploring, navigating, turning their heads around. Yeah, as you can see from the barcode, we have one very persistent structure in H0, the Betty number 0, and as well as one persistent Betty 1 feature. And that basically says, so there's a one entity in this space and the one-dimensional whole, which basically describes the existence of a ring. And and the rest of the plot basically shows that we can fit a spline to depict this backbone, and then parameterize it using a variable, and then read out the values for all the points in the space. Of course, we can try to decode the actual head angle from this parameterized variable that's discovered in an unsupervised way. And it turns out the decoding actually tracks the actually observed head direction, as well as a decoding from supervised method very well. And it also matches the tuning. Most amazing thing, I think, is that it's time resolved, meaning the relative distance between different head directions are the same as the relative distance among the different states, like in activity space, depicted by this parameterized alpha. Now we have the topological structure, and the authors wanted to ask, like, if this structure is autonomously generated and can be stabilized and self-sustained. What do they mean by that? You know, during awake, the animal is constantly bombarded by sensory input, and it is likely that the structure we saw was maintained by those sensory input, so. However, for autonomously generated and self-sustained structure, and it should remain the same even without those input. So that's why the authors turned into the sleep period, when the animals certainly don't get any useful sensory input, and see how these neurons may change their activity. So they perform the same SPUD, the spine parameterization for unsupervised decoding method, to the REM sleep data. REM stands for rapid eye movement. And what they found is that the manifold structure very well aligns with that during awake. But to really address the questions I was talking about, the authors measured the flux vectors at different points in the space. So flux vectors describe the change of activity. And they found that on the manifold or close to the manifold, and the length of the vector are really typically short, and the direction is along those manifolds. And away from this manifold, away from this ring, and the direction is usually the activity is usually being pulled towards the manifold, but it also has a component that goes along the direction of the manifold. And so furthermore, the author hypothesized, or the author asked, are the different states in this manifold are energetically equivalent? So what that means? So just imagine a table, a flat table, and we put a ball on it, and the ball is not going to roll to any direction. But if the table is shaking in a random way, then that creates some noise to the ball, and the ball may roll to the left or roll to the right equally, and resulting in some sort of random walk on the table. If the table is being shaken more vigorously, then you can imagine a more rapid random walk. If the shaking makes the table slanted, so then that provides a correlated input to the ball, and the ball will drift in certain direction. So what they found is that indeed the manifold, different states along the manifold are energetically equivalent, as shown by the fact that in the top plot, given an alpha, the change of alpha is about 0. It doesn't mean there's no movement, but it actually means the movements toward different directions cancels out with each other. Also, it's shown in the lower plot that the likelihood of the state present in a given alpha is roughly the same across different alphas. Now let's look a little deeper into the behavior of states, the change of states, in terms of how the state diffuses during sleep. So if we look at the awake data, because during exploration, the change of head direction is often correlated in a short period of time. For example, if I'm trying to look to my right, then for a period of time I'm turning my head to the right, and rather than just doing some kind of random jittering and gradually drifting my head to the right. But during REM sleep, the temporal correlation is much weaker. So that's why you see in the inside of this plot, and we have a hyperlinear or quadratic relationship between the squared change in distance against time. So that reflects the presence of some correlated input. But if we look at the REM sleep data, then you see that it's much closer to a linear relationship. That suggests that the noise that's contributing to the dynamics is a lot less correlated than those during awake. However, if the authors build a model, it's called a continuous attractor model, they're only considering the noise that's internal to the neurons, for example, from the Poisson, the stochastic Poisson process of spike generation, or there's some random external input that gives rise to uncorrelated noise to this population of neurons. Then you see a very weak drifting in terms of the slope. But the actually experimentally observed slope is much steeper. And in order to reproduce that steep slope, then the authors had to incorporate a velocity noise into this model. And the author said, so this input must be low dimensional and is consistent with what the animal experienced during awake. It's consistent with the sensory input of head direction change that the animal experienced during awake. So sleep is generally divided into two kinds of states. So one is called REM sleep, which was what we were talking about, and another is called non-REM sleep, or a non-rapid eye movement sleep. It's also commonly known as slow wave sleep, characterized by the kind of slow changing, oscillating, fluctuating dynamics as a result of correlated neuronal firing across the populations. So in non-REM sleep or slow wave sleep, in the hippocampal circuits, we have the famous replay event. Basically, the activity of the neurons actually recapitulate or replay what people recorded or what the animal experienced during awake time. This is called hippocampal replay. However, in the head direction circuit, it hasn't been reported about any replay events. And the authors here have suspected that it might be due to the specific analysis method. Again, they performed the SPUD and got this structure. So this is no longer a one-dimensional ring manifold. Rather, it's a two-dimensional cone-shaped structure. And so this we know that from the Betty barcode. So different from before. Well, same as before. We have a Betty, one persistent structure in H0, but we no longer have this persistent structure in H1, which describes a one-dimensional whole. If we compare the awake manifold with this non-REM sleep manifold, the ring in awake manifold really traces the rim of this cone structure. The authors parameterized this cone shape with two directions. One is the tangential direction and another is the radial direction. And it turns out like the tangential direction still encodes the head direction as that in the awake case. However, the radial structure reflects the population activity, which is the characteristics of the slow wave sleep, the non-REM sleep. So this is the shape of this low dimensional manifold. But what about the dynamics? What about the diffusions? So here the authors observed the big sweeps along the manifold, and the sweeps are coherent, meaning for a motion in one direction typically continues or persists in that direction, as shown by the autocorrelation in this plot. It's shown in the color red. And in comparison, we can see similar autocorrelations during awake, but much weaker autocorrelations during REM sleep, as we have talked about. As a result, the curve characterizing the diffusion shows a quadratic shape rather than a linear shape, and as similar to the awake period. Interestingly, the trajectory in terms of the head direction decoding, it travels 8 times faster as the head direction decoding during awake period. And the factor of 8 is an interesting one, because it's roughly the same as how the hippocampus may replay the positions of the animal traveling in space during awake than during the slow wave sleep. In addition, those sweeps occur in some transient activity in LFP, which corresponds to or which is correlated to sharp waves activity. Okay, so that's it. That's the last figure, but this is a very rich paper, and I really encourage you to go in there and read in more details, and there are a lot of interesting data in the supplemental information, as well as a nice read of the supplemental mathematical notes, and I really enjoyed it. So in the end, instead of a summary recapping all the result, reiterating all the result of this paper, I just want to mention like something I did in my project. I was dealing with sequences of sensory motor control. So in a side project that Natasha took on, so we first trained the animal in awake about this sensory motor sequences, and then put the animal to sleep and record from the same cortical brain regions, and want to see how the neurons in there may be reactivated in a way that's relatable to the awake state. And I think this is a perfect opportunity, or with or without the sleep experiment, it's a perfect opportunity to apply this method to my data. But with the sleep data, it's become more interesting to see how the diffusion may go and what is the dynamic, what structures we see here. And I don't expect it to be a one-dimensional ring, because there are obviously more than one important variable for this behavior. Yeah, but anyway, I guess the first thing I would do is to contact the authors and ask for some code to see if I'm able to get it running, to understand and get it running, and couldn't wait to see what turns out in my data. And furthermore, we're going to have some exciting new recording technology going in our lab, and we'll be able to record a lot more neurons simultaneously in probably more than one region of interest in the brain, and it can be done probably cortically, and so we can get the data from both awake and sleep, a lot of neurons in awake and sleep, and who knows what kind of neuronal structure, what kind of activity structures we're going to have. So that concludes this journal walk, and I'm at the lab now. Yeah, see you next time.